Water pollution is the contamination of water bodies such as lakes, rivers, oceans, ponds, streams, and groundwater. The uses we make of water in certain water bodies is greatly influenced by the quality of water found in them. Activities such as fishing, swimming, boating, shipping, and waste disposal have very different requirements for water quality. Water of a particularly high quality is needed for potable water supplies. And since clean water is now considered a scarcity, water quality management is concerned with the control of pollution from human activity so that the water is not degraded to the point that it is no longer suitable for intended uses. There are many sources of water pollution, but the two main sources are called point sources and non-point sources. Point sources refer to the pollutants that belong to a single source, meaning the pollutants are released from discrete conveyances such as discharge pipe and are often regulated by the federal and the state agencies. Domestic sewage and industrial waste are considered as point sources. Non-point sources are opposite of point sources. Non-point sources are a combination of pollutants from a large area rather than specified identifiable sources such as discharge pipes. So, the pollutants present in the water flows over the surface of the land or along natural drainages or channels to the nearest water body. Runoff is generally associated with non-point source, as water is emptied into streams and rivers after accumulating contaminants from sources like garden, parking lot, or construction sites. Actually, runoff from agricultural land is considered as a significant non-point source because agricultural runoff carry nutrients, toxic organic compounds, and chemicals from pesticides and fertilizers that may pollute the water. Much of non-point sources occurs during rainstorms and spring melt, resulting in large flow rates that make treatment even more difficult. Oxygen-demanding material is another source for water pollution. Anything that can be oxidized in the receiving water body with the consumption of dissolved molecular oxygen is termed oxygen-demanding material. This material is usually biodegradable organic matter, but also includes certain inorganic compounds. The consumption of dissolved oxygen, or DO, poses a threat to any form of aquatic life that requires oxygen to live. The critical level of DO varies greatly among species. Say for example, brook trout may require 7.5 milligrams per liter of DO, while carp survives at 3 milligram per liter of DO. So almost any naturally occurring organic matter, such as animal droppings, crop residues, or leaves that get into the water from non-point sources contribute to the depletion of dissolved oxygen. The point sources also contribute to the depletion of DO, specifically the domestic sewage, mainly from human waste and food residue. Nutrients are needed for the growth of aquatic life. However, some wastewater and sewage contain high levels of nutrients, which is not considered as a good thing. Too much nutrients in the water encourages the growth of algae in the water, which disturbs the food web. Once the algae die, they turn into an oxygen-demanding material and other organisms might die because of oxygen starvation. This makes the nutrients as a source of water pollution. So, phosphorus-based detergents, fertilizers, and food processing waste are some major sources that contributes to nutrient pollution. Pathogenic organisms are also a source of water pollution. Microorganisms found in wastewater include bacteria, viruses, and protozoa excreted from deceased persons or animals. These pathogens may also come from the feces and stool of a human being or animals. So, when the wastewater contaminated with pathogens is discharged into surface water, they make the water unfit for drinking. So, these microorganisms may cause fishes and other aquatic life forms to die because of toxicity. Pathogen contamination can also cause serious illness to humans who drink from surface waters. This illness includes hepatitis, cholera, E. coli, and typhoid. Another source for water pollution are suspended solids, organic and inorganic particles that are carried by the wastewater into a receiving water are termed suspended solids or SS. So some particles do not easily dissolve in the water. They will settle at the bottom of the water body, which may pose threats to the aquatic life living in the floor of the water body. Organic suspended solids may also exert an oxygen demand, while inorganic suspended solids are discharged by some industries but result mostly from soil erosion, which is particularly bad in areas of logging, strip mining, and construction activity. Salts are also considered as a source of water pollution. The salts and other things that don't evaporate are called total dissolved solids or TDS. Salts are present in any water body but vary in concentration. When there is a high concentration of salt in the fresh water, it poses threat to the natural population of plants and animals, and the water may no longer useful for irrigation and any public use. High concentrations of salts are discharged by many industries, and the use of salts on roads during the winter causes high salt levels in urban runoff, especially during the spring snowmelt. Another source for water pollution include pharmaceuticals and personal care products or PPCPs. So these are a class of compounds that are applied externally or ingested by humans, pets, and other domesticated animals. They are released to the environment through the disposal of expired, unwanted, or excess medications to the sewage system by individuals, pharmacies, or physicians. Personal care products or PCPs, such as hand and dish soap, oral hygiene products, and air fresheners may contain compounds such as phthalates, phenols, and tricloxan. Tricloxan is also considered a registered pesticide. Actually, there are found seven carcinogenic compounds in PCPs. This includes hydroquinone, ethylene dioxide, formaldehyde, nitrosamine, acrylamides, and polycyclic aromatic hydrocarbons. Endocrine disrupting chemicals can also pollute the water. The class of chemicals known as endocrine disruptors or EDCs alter the normal physiological function of the endocrine system and can affect the synthesis of hormones. 17-beta estradiol is a naturally occurring EDC. It is a naturally occurring estrogen used in birth control that is discharged into the wastewater. Polychlorinate biphenyls are EDCs used commonly in pesticides. EDCs can mimic estrogens, androgens, or thyroid hormones or their antagonists, although the structure of many EDCs bear little resemblance to that of natural hormones that which they interfere. Pesticide is also considered a source of water pollution. The major classes of pesticides are herbicides, insecticides, and fungicides. The presence of pesticides in the surface water may pose threat to any form of aquatic life. This is because certain chemicals present in pesticides like cypermethrin and neonicotinoid are considered as toxic to certain aquatic life forms. So the presence of pesticides in surface water is mostly due to agricultural runoff. So pesticides coming in contact with surface water may be dissolved or mixed physically with water, which causes surface water pollution. Moreover, pesticide can also reach the groundwater. The applied pesticides are being washed deep into the ground by rainwater that may go deeper underground and into wells. This may cause contamination to drinking water, which comes from wells and hand pumps. Another source of water pollution is heat. Although heat is not often recognized as a pollutant, those in the electric power industry are well aware of the problems of disposing of waste heat. The water being released by many industries is much hotter than the receiving water. This increase in temperature may be beneficial to certain water bodies where clams and oysters are present, but other forms of aquatic life only thrive in cool water. High temperatures also increase the rate of oxygen depletion in areas where oxygen demanding wastes are present. Water pollution in rivers. The impact of pollution on a river depends both of the nature of the pollutant and the unique characteristics of the individual river. Some of the most important characteristics include the volume and speed of water flowing in the river's depth, the type of bottom, and the surrounding vegetation. Other factors include the climate of the region, the mineral heritage of the watershed, land use patterns, and types of aquatic life in the river. Water quality management for a particular river must consider all these factors. Thus, some rivers are highly susceptible to pollutants such as sediment, salt, and heat, while other rivers can tolerate large inputs of these pollutants without much damage. Total maximum daily load or TMDL. So TMDL is established for the impaired waters. Impaired waters meaning waters that do not meet the water quality standards that the states, territories, and authorized tribes have established for them. So a TMDL specifies the maximum amount of pollutant that a water body can receive and still meet water quality standards. It allocates pollutant loadings that may be contributed among point and non-point source. So the TMDL is defined as the shown in the picture below. When oxygen-demanding material is introduced in any water body, it causes oxygen depletion, which may pose threat to the aquatic life forms if the concentration of oxygen fall below critical point. Organic oxygen-demanding materials are commonly measured by determining the amount of oxygen consumed during degradation in a manner approximating degradation in natural waters. The amount of oxygen required to oxidize a substance to carbon dioxide and water may be calculated by stoichiometry if the chemical composition of the substance is known. This amount of oxygen is known as the theoretical oxygen demand or THOD. In contrast to THOD, the chemical oxygen demand, or COD, is a measured quantity that does not depend on knowledge of the chemical composition of the substances in the water. In the COD test, a strong chemical oxidizing agent, chromic acid, is mixed with a water sample and then boiled. The difference between the amount of oxidizing agent at the beginning of the test and that remaining at the end of the test is used to calculate the COD. If the oxidation of an organic matter is carried out by microorganisms using the organic matter as a food source, the oxygen consumed is known as the biochemical oxygen demand or BOD. The actual BOD is less than the THOD due to the incorporation of some of the carbon into the new bacteria cells. BOD, COD, and THOD will only be equal under rare circumstances. While the ultimate BOD increases in direct proportion to the concentration of the degradable organic matter, The numerical value of the rate constant is dependent on the following: the nature of the waste, the ability of the organisms in the system to utilize the waste, and the temperature. Nature of waste. There are literally thousands of naturally occurring organic compounds, not all of which can be degraded with equal ease. Simple sugars and starches are rapidly degraded and will therefore have a very large BOD rate constant. The BOD rate constant for a complex waste depends very much on the relative proportions of various components. Ability of organisms to utilize waste. Any given microorganism is limited in its ability to utilize organic compounds. As a consequence, many organic compounds can be degraded by only a small group of microorganisms. Temperature. Most biological processes speed up as the temperature increases and slow down as the temperature drops. Because oxygen utilization is caused by the metabolism of microorganisms, the rate of utilization is similarly affected by temperature. Therefore, BOD rate constant should be experimentally determined for the temperature of the receiving water. But there are problems encountered concerning temperature. Since the temperature of the receiving water changes throughout the year, large number of tests would be required to define K. An additional difficulty is the task of comparing data from various locations having different temperatures. And the solution is laboratory testing is therefore done at a standard temperature of 208 degrees Celsius and the BOD rate constant is adjusted to the receiving water temperature. There are a lot of methods that may be used to determine K and the oxygen equivalent of organic compounds at time zero, from an experimental set of BOD data, but the simplest yet least accurate is to plot BOD versus time. The rate equation is used to solve for K. One simple method around this is called Thomas graphical method. So the procedure for determining the BOD constant by Thomas graphical method is as follows. In order to have as much in order to have as much consistency as possible, it is important to standardize testing procedures when measuring BOD. So here are the procedures. Step 1. A special 300 ml BOD bottle is completely filled with a sample of water that has been appropriately diluted and inoculated with microorganisms. So a stopper was used to exude air bubbles from the bottle. The BOD of the diluted sample is expected to be between 2 to 6 mg per L since the amount of oxygen that can be dissolved is 9 mg per L. Samples are diluted with a special dilution water that contains all of the trace elements required for bacterial metabolism so that degradation of the organic matter is not limited by the lack of bacterial growth. The dilution water also contains an inoculum of microorganisms so that all samples tested on a given day contain approximately the same type and number of microorganisms. The ratio of the undiluted to diluted sample is called the sample size, usually expressed as a percentage, while the inverse relationship is called the dilution factor. The appropriate sample size to use can be determined by dividing 4 mg per L, the midpoint of the desired range of diluted BOD, by the estimated BOD's concentration in the sample being tested. A convenient volume of undiluted sample is then chosen to approximate the sample size. Step 2. Blank samples containing only the inoculated dilution water are also placed in BOD bottles and stoppered. Blanks are required to estimate the amount of oxygen consumed by the added inoculum of microorganisms called seed in the absence of the sample. Step 3. The stoppered BOD bottles containing diluted sample and blanks are incubated in the dark at 208 degrees Celsius for the desired number of days. 5 days is the standard time, but to determine the ultimate BOD and the BOD constant rate, additional time is required. As mentioned earlier, the BOD test is conducted at a standard temperature of 208 degrees Celsius so that the effect of temperature on the BOD rate constant is eliminated, and results from different laboratories can be compared. Step 4. After the desired numbers of days has elapsed, the samples and the blanks are removed from the incubator, and the dissolved oxygen concentration in each bottle is measured. The BOD of the undiluted sample is then calculated using the following equation. Additional notes on biochemical oxygen demand. Although the 5-day BOD has been chosen as the standard value for most wastewater analysis and for regulatory purposes, the ultimate BOD is actually a better indicator of total waste strength. So if you're wondering why the 5-day BOD was chosen as the standard value for most purposes, it is because the test was devised by the environmental engineers in England, where rivers have travel times to the sea of less than 5 days, so there was no need to consider oxygen demand at longer times. Additionally, for different types of waste having the same BOD5, the ultimate BOD is the same only if, by chance, the BOD rate constant are the same. Oxygen consumption due to oxidation of the carbon is called the carbonaceous BOD or CBOD, while the oxygen consumption due to nitrogen oxidation is called nitrogenous BOD or NBOD. The organisms that oxidize the carbon in organic compounds to obtain energy cannot oxidize the nitrogen in these compounds. Instead, the nitrogen is released into the surrounding water as ammonia, which is in the form of ammonium cation at normal pH values. So nitrification is a process by which the ammonia released from organic compound and from other sources is oxidized to nitrate by special nitrifying bacteria as their source of energy. The figure shows the BOD curves showing both carbonaceous and nitrogenous BOD. The rate at which the NBOD is exerted depends heavily on the number of nitrifying organisms present. The figure is the typical Streeter-Phelps DO sag curve. DO sag curves give us the characteristic response in oxygen levels as a result of discharging oxygen-demanding waste to river or stream. The figure shows the oxygen sag downstream of an biodegradable organic chemical source. So, a simplified mass balance helps us understand and solve the DO sag curve problem. This is the conservative mass balance diagram for DO mixing. In mass balance approach, the product of the water flow and the DO concentration yields a mass of oxygen per unit of time. The mass of DO in the river after mixing equals the sum of the mass flows. In similar fashion for ultimate BOD. Oxygen deficit is the amount by which the actual dissolved oxygen concentration is less than the saturation value with respect to oxygen in the air. So the picture shows the expression for oxygen deficit. The beginning of the DO sag curve is at the point where a waste discharge mixes with the river. The initial deficit is calculated as the difference between the saturated DO and the concentration of the DO after mixing. The two figures are the mass balance diagram of DO in a small reach of river and a simplified mass balance of Streeter-Phelps model. This is the Streeter-Phelps DO sag equation. The deoxygenation rate constant differs from the BOD rate constant because there are physical and biological differences between a river and a BOD bottle. So in 1966, Bosco has developed a method of estimating the deoxygenation rate constant from the BOD rate constant using the characteristics of the stream. Reaeration is an important natural process where rivers are used for the disposal and partial treatment of waste. So reaeration is the exchange of gases between the atmosphere and the water. This process operates to maintain oxygen near the saturation concentration. The value of reaction rate constant depends on the degree of turbulent mixing, which is related to stream velocity, and on the amount of water surface exposed to the atmosphere compared to the volume of water in the river. So, a narrow, deep river will have a much lower reaction rate constant than a wide, shallow river. O'Connor and Dobbins, 1958, developed a generalized empirical equation to estimate the reaeration rate constant based on the characteristic of the stream and the molecular diffusion of oxygen into the water. The speed of the flow in the river and the distance downstream, the travel time may be estimated from the basic relationship between time, distance, and speed. So time is equivalent to distance over speed. To relate travel time to a physical distance downstream, one must also know the average stream velocity. The lowest point of the DO sag curve and is of major interest since it indicates the worst conditions in the river is the critical point. The time to the critical point, T subscript C, can be found by differentiating Streeter-Phelps DO sag equation, setting it equal to 0 and solving for T using base E values for the reaction rate constant and the oxygenation rate constant. TMDL mass loading or TMDL calculations based on mass loading. To use the DO sag equation as presented here, the mass loading of ultimate BOD must be converted to a concentration. Following our general approach for calculating concentration from mass flow, we divide the mass flow by the flow of the water carrying the waste. Then the mass discharge units are then converted to mg per day and the water flow rate to liter per day so that the days cancel. The beginning point for water quality management in rivers using the DO sag curve is to determine the minimum DO concentration that will protect the aquatic life in the stream. This value, called DO standard, is generally set to protect the most sensitive species that exist or could exist in the particular river. Using the DO sag curve to determine the adequacy of wastewater treatment, it is important to use the river conditions that will cause the lowest DO concentration. In many cases, nitrogenous BOD or NBOD has at least as much impact on dissolved oxygen levels. In most wastewater plants, they produce a typical BOD effluent that also contains approximately 300 mg per L of nitrogen, which would mean an NBOD of about 137 mg per L if it were discharged as ammonia. The classical DO-SAG curve assumes that there is only one point source discharge of waste into the river. However, in reality, there are multiple sources. So rapids affects the DO levels. Especially in small rivers, it plays a major role in maintaining high DO levels. Then some rivers contain large deposits of organic matter in the sediments. So when the matter decays, it becomes an oxygen-demanding material, affecting the DO levels. Then aquatic plants can also have a substantial effect on DO levels. Aside from the oxygen-demanding material waste, nutrients can also contribute to deteriorating water quality in rivers by causing excessive plant growth. So nutrients are those elements required by the plants for their growth. So they include carbon, nitrogen, phosphorus, and a variety of trace elements. Plant growth may be desirable, but excessive plant growth can create a number of undesirable conditions, such as thick slime layers on rocks and dense growth of aquatic weeds. In many rivers, turbidity and strong water currents prevent plant growth. So these are the three reasons why nitrogen is detrimental to the receiving water body. The effect of phosphorus. The major deleterious effect of phosphorus is that it serves as a vital nutrient for the growth of algae. Management strategy. The practical control of nutrient-caused water quality problems in streams is based on removal of nitrogen and/or phosphorus from wastewaters before they are discharged and using best management practices or BMPs to minimize nutrient in agricultural runoff. So the pollutants that contribute to water pollution in lakes are oxygen-demanding waste, pathogens, toxic chemicals from industrial discharges, and phosphorus, the most dominant pollutant in lakes. Stratification and turnover of lake. So stratification is when the lake is divided into three distinct layers, namely the epilemnon, hypolemnon, and metalemnon. Epilemnon, the shallowest layer or the warm surface layer. Hypolemnon, the deepest layer, and it is cold and dense. And then the metalemnon is the middle layer or the transition zone between the warm epilemnon and the cold hypolemnon. And turnover is a phenomenon whereby the entire volume of the lake is mixed by the wind. So nearly all lakes in the temperate climatic zone become stratified during the summer and overturn in the fall due to changes in the water temperature that result from the annual cycle of air temperature changes. Lakes in cold climates undergo winter stratification and spring overturn as well. Lakes contain several distinct zones of biological activity. Lake contains several distinct zones of biological activity, largely determined by the availability of light and oxygen. So the most important biological zones are euphotic, littoral, and benthic zone. So the euphotic zone is the upper layer of water through which sunlight can penetrate. In euphotic zone, plants produce more oxygen by photosynthesis than they remove by respiration. Then the littoral zone, or the shallow water near the shore in which rooted water plants can grow. The extent of the littoral zone depends on the slope of the lake bottom and the depth of the euphotic zone. Thus, this zone cannot extend deeper than the euphotic zone. Then the zone at the bottom of the lake is called benthic zone. The bottom sediments comprise the benthic zone. Once organisms living in the overlying water die, they will settle at the bottom and decompose by organisms in the benthic zone. Bacteria are always present. The presence of higher life forms such as worms, insects, and crustaceans depends on the availability of oxygen. The productivity of the lake is a measure of its ability to support food web. It is determined by measuring the amount of algae growth that can be supported by available nutrients. Although a more productive lake usually will have a higher fish population, the number of most desirable fish may decline. In fact, increased productivity generally results in reduced water quality because of undesirable changes that occur as algal growth increases. So there are four lake classifications: the oligotrophic lake, eutrophic lakes, mesotrophic lakes, and senescent lakes. So oligotrophic lakes have a low level of productivity due to a severely limited supply of nutrients to support algal growth. So as a result, the water is clear enough that the bottom can be seen at considerable depths. Then the opposite of oligotrophic lakes are eutrophic lakes. Eutrophic lakes have a high productivity because of an abundant supply of algal nutrients. The algae cause the water to be highly turbid, so the euphotic zone may extend only partially into the epilemnion. Highly eutrophic lakes may also have large mats of floating algae that typically impart an unpleasant taste and odors to the water. Then lakes which are intermediate between oligotrophic and eutrophic are called mesotrophic. Although substantial depletion of oxygen may have occurred in the hypolemnion, it remains aerobic. Then lastly, the senescent lakes. These are very old, shallow lakes which have thick organic sediments and rooted water plants in great abundance. These lakes will eventually become marshes. Algae growth requirements. So for algae to grow, all nutrients must be available. So this includes carbon, nitrogen, phosphorus, and trace elements. So lack of any nutrient will limit the total algal population. So first, carbon. Algae obtain their carbon from carbon dioxide dissolved in the water. When algae are either consumed by higher organisms or die and decompose, the organic carbon is oxidized back to carbon dioxide, which returns either to the water or to the atmosphere. Then nitrogen. Nitrogen in lakes is usually in the form of nitrate and comes from external sources by way of inflowing streams or groundwater. Denitrification reduces the average time nitrogen remains in the lake system. So the reaction for denitrification is shown below. Then the third nutrient is phosphorus. Phosphorus in lakes originates from external sources and is taken up by algae in the inorganic form and incorporated into organic compounds. Then lastly, trace elements. The quantities of trace elements required to support algal growth are so small that most freshwaters have sufficient amounts of for a substantial algal population. Liebig's law of minimum states that the growth of a plant is dependent on the amount of foodstuff that is presented to it in minimum quantity, meaning that the limiting nutrient is the foodstuff that is presented to it in the minimum quantity. So when this law is applied to the growth of algae, phosphorus is deemed to be the limiting nutrient, since it is the only one not readily available in the atmosphere. Because phosphorus is usually the limiting nutrient, control of cultural eutrophication must be accomplished by reducing the input of phosphorus to the lake. Once the input is reduced, the phosphorus concentration will gradually fall as phosphorus is buried in the sediment or flushed from the lake. So, to be able to reduce phosphorus inputs, it is necessary to know the sources of phosphorus and the potential for the reduction. So, the most important sources are municipal and industrial wastewaters, seepage from the septic tanks, and agricultural runoff that carries phosphorus fertilizers into the water. Municipal and industrial wastewaters. All municipal sewage contain phosphorus from human excrement. The only effective way of reducing phosphorus is through advanced wastewater treatment processes. Then the next one are septic tank sewage. The shores of many lakes are dotted with homes and summer cottages, each with its own septic tank and tile field for waste disposal. As treated wastewater moves through the soil toward the lake, phosphorus is absorbed by soil particles, especially clay. To prevent phosphorus from reaching the lake, it is necessary to put the tile field far enough from the lake that the absorption capacity of the soil is not exceeded. Then agricultural runoff. Because phosphorus is a plant nutrient, it is an important ingredient in fertilizers. It can be introduced to the water body through rain or soil erosion. Waste, minimi- soil erosion. Waste minimization can be applied to the control of phosphorus loading to lakes from agricultural fertilization by encouraging farmers to fertilize more often with smaller amounts and to take effective action to stop soil erosion. Acidification of lakes is the process by which the pH level of lakes is reduced. So, pure rainwater is slightly acid. Acid rain is usually defined to be a precipitation with a pH less than 5.6. Then, acidification of water releases high toxic aluminum cation to the water. However, most lakes are buffered by carbonate buffer system. Other factors that affect the susceptibility of lake to acidification are the permeability and the depth of the soil, the bedrock, the slope and size of the watershed, and the type of vegetation. An estuary is formed along the coastline where fresh water from rivers and streams flow into the ocean. It is a place of transition where fresh water mixes with salt water. Estuaries are influenced by tides. Like other surface waters, estuaries are subject to a plethora of pollutants. Although the technologies and policies that serve to manage the water quality of lakes and rivers are also applied to estuaries, the management of the water quality is complicated by the number of political jurisdictions that touch on the estuary as well as the competing needs of organisms that live there. Groundwater, by its very location, has a large measure of protection from contaminants that are found in surface waters. However, once groundwater becomes contaminated, its location and low rate of replacement with fresh water makes it difficult to return into a pristine state. So the two major sources of contaminants are of concern: uncontrolled releases of biological chemical contaminants and saltwater intrusion from overjumping of wells. Freshwater aquifers near oceans or above saline aquifers may become contaminated with saltwater when water is pumped from wells that draw water that is too close to the freshwater saltwater interface. So using the notation in figure 720 shown below, and assuming that fresh water moves horizontally to the ocean and that the interface is abrupt and that it occurs at the shoreline, we can express the freshwater pressure at any point of the interface as shown below. When fresh clean water is underlain by saline water, pumping a well in the fresh water will cause the interface to rise below the well. The coning is in response to the pressure reduction in the interface that results from the drawdown of the water table around the well. So the height of the upconing below the center of a well after an infinite pumping time at the freshwater will may be estimated using the Beer and Dagan 1968 equation as modified by Schmorak and Mercado 1969. A critical point in the relationship between the upconing and the drawdown is reached when the saltwater cone height reaches approximately 0.4 L to 0.6 L. And when the cone height exceeds this critical height, the cone may jump to the bottom of the well. Thus, when fresh water is underlain with saline water, prediction of the upconing is important to prevent saltwater intrusion.